Hello everyone, my name is Anna Hartikainen and as Lena mentioned, I just defended my PhD and I'm working in Reetta Satokari's group, and today my topic is microbiome changes in FMT. So let's start. So here is the outline for today. So first I will tell you something about the background, then about the microbiome changes in 3 different conditions, recurrent C. difficile infection, irritable bowel syndrome, and then inflammatory bowel disease. And then in the end have some kind of a question that do we need the microbiome changes and which kind of changes to have the clinical effect and what do we know at the moment about that. So let's start. So many of you know that we are living with the microbes, of course, and mainly they are living in our guts and they are bacteria. But of course, we have different microbiomes all over our body, for example, in mouth, skin, vagina, also in lungs and so on. So microbiota is used as a term to describe the microbes that live in a certain environment, and microbiome is this kind of a collective term for the microbes, their genes, and the environment that they live in. And in biology, we like to classify things. So here on the right, you can see a taxonomical classification. And for example, this bacterium Lacticacea bacillus, that's its genus. Species is Rhamnosus, and bacteria also have different strains. So this one is GG, and actually it is used also as a probiotic. And then how do we study the microbiome? Just a few words about that. When we are talking about the gut microbiome, usually we are the starting material is actually feces because of the non-invasive collection method. And of course, then the cons for that is that we only know what is coming out from the system, not what's happening in the system. But anyway, it is the most common sample that we use. And traditionally, the bacteria have been cultivated on a petri dish, and then identification has done with that. But that's probably not the most efficient way to study the whole microbiota, especially the gut microbiota, because of the conditions in the gut. For example, some bacteria might have very strict growth conditions. For example, some bacteria need the anaerobic conditions. So what we can do is to do the DNA extraction for the whole sample and then do sequencing. And here are 2 different sequencing methods that are commonly used. And the first one is 16S rRNA gene sequencing, where one gene is amplified, then that is used to make the identification. And basically this method can answer to the question which bacteria are in the sample. And another one is shotgun metagenomics, where all DNA in the sample is sequenced. And with this method, actually, we can have more resolution, so we can go down to the species and even strain level. And also we have 2 different methods for this. We can have the reference-based methods and also different kind of methods that we don't need the reference, so we can also identify new species and strains. And with this method also we can know something about the functions that the microbes have. And as Joshbert already nicely told, we have very different FMT strategies used. And here I have highlighted the standard procedure for recurrent C. difficile infection in Finland. So sometimes the antibiotics are used for the underlying conditions, but sometimes they are also used as a pre-treatment to decolonize the gut. And the bowel lavage is used to empty the colon and the whole system from the fecal material. And this is used especially when the FMT is given by a colonoscopy. But also we can give the FMT with the upper route via gastroscopy, capsules, or even as a drink. And also we have different methods. Do we give the FMT once or then multiple times? And the amount is usually 30 to 60 grams, but it also can be more. As Joshbert also told, nowadays the universal donor is used mostly because then we can give the transplant to many patients. And sometimes multiple donors are used to increase the diversity of the bacterial diversity in the fecal transplant. And already Jasper also mentioned that the transplant itself can be given fresh or frozen, and it can be handled in anaerobic or aerobic conditions. And definitely all of these strategies and those choices that we make, they might have effect on the bacterial colonization or then to the clinical outcome. And of course, it's hard to compare the studies if the strategies are very different, but then also with the very different indications that we use FMT, we might need different strategies. Okay, then I will go shortly through the 3 different indications that I'm going to talk next. So, of course, this recurrent C. difficile infection is caused by this one bacterium that has these spores that can then terminate if the bacterial composition in the gut has been disrupted, for example, by antibiotics, and these spores are left intact, and then this vicious cycle of recurrent infections may start. And as we heard, FMT is very effective to treat this disease. Then irritable bowel syndrome, it has been known as dysfunctional gastrointestinal disorder. Nowadays, it is referred as a disorder of the gut-brain axis, which means that there is this dysregulation of the signals between the gut and brain, and then symptoms are localized to the gut. And FMT is also studied in this regard. And inflammatory bowel disease and pouchitis. Pouchitis is a kind of under this umbrella. These are chronic inflammatory diseases in the gut, and even though we don't know the etiology, we know that there is a genetic and environmental component, and also impaired immune system regulation towards the gut microbiota. And especially with the ulcerative colitis patients, some patients may need surgical interventions where the colon, for example, is removed and this J pouch is constructed. This is very, of course, efficient treatment for those patients. However, also this pouch may be inflamed, and this is called then pouchitis. So let's go to the microbiome changes. So we know that because of the very vicious infection, and then of course the use of antibiotics, the recurrent C. difficile infection patients, they have very disrupted microbiota. Here in the panel A, you see average relative abundance of different taxa in these patients. And the more similar these bar plots are, the more similar is the microbiota composition. And here, before FMT, we can see that the microbiota is very different from the donors. But very early on, at 3 days time point, we can see that it starts to resemble more of that of the donors, and the effect actually lasts up to 12 months. Also here in the PCOA plot, we can see the same effect. So every point of this picture, they represent one patient or sample. And here before FMT, these samples are clustered together. And after FMT, we can see that they are clustered with the donors, meaning that the microbiota compositions are more similar to each other. As already mentioned, FMT can be given also with different routes. So here, Gao et al., they have looked at the oral capsules used and then the colonoscopy in recurrent C. difficile infection, and the same effect can be seen here. So the microbiota is different before FMT, and then after FMT, we can see that the microbiota clusters with the donor. So the microbiota becomes more similar with the donor. So all in all, in recurrent C. difficile infection, we see this very high donor colonization and microbiota change. Okay, then with irritable bowel syndrome, it's more trickier because that is more multifactorial disease, and also the clinical trial outcomes have been more variable. Also, the microbiome changes have been more variable. So here, Gol et al. Show that these patients who have had effect on the treatment, they actually showed this kind of shift in microbiota towards the donor. However, those patients who did not have any effect to the treatment, they didn't have the similar kind of shift towards the donor. So these patients had increase of bacteroidetes and decrease of firmicutes, for example. Also here, this El Salhi et al. Paper, they showed that the clinical effect and also the microbiota changes can be very long-lasting. Here you can see where they used 30 grams of the fecal transplant and 60 grams of the fecal transplant, that the baseline samples in both cases are clustered separately from the 2-year and 3-year follow-up time points. Also in this paper, they found 10 bacteria that had significant correlations with the IBS symptoms and fatigue after FMT treatment. We also studied IBS patients in FMT clinical trial. However, we didn't see significant differences in the symptoms between the placebo and FMT group, but we saw a transient symptom relief in the FMT group. But we also saw a very drastic change in the FMT-treated patients' microbiota. And here, for example, the family Prevotellacea increased significantly in these FMT-treated patients, and also the bacterial richness increased in these patients. However, in our clinical trial, we couldn't see any significant differences between the non-responders and responders to the treatment seen, for example, here with the richness and also with the microbiota composition and the potential functionality. We couldn't see significant differences between these groups. We were able to reach the strain and species level in this study, and when we looked more further, we could see that those patients who had FMT, they were actually dominated with the Prevotella copri bacterium, and it was very prevalent in these patients. However, we also saw that the baseline microbiota composition, so this niche availability, had an effect on the colonization of this strain, where we see that these patients who had high Prevotella copri abundance at baseline, they were dominated with their own strain throughout the whole 1-year follow-up. However, these patients who did not have that high Prevotella copri at baseline, they were clustered with the donor and dominated with the donor strain. So, as already mentioned, this donor-patient compatibility plays a role in FMT, definitely. Okay, so then something about inflammatory bowel disease, which is also studied in FMT very much. So I think this is the same study that Jospert also mentioned, where one donor was actually more successful than the other. And also in this regard, they showed that the Prevotellacea dominant clustered patients, they actually were associated with the non-response to the FMT treatment. And for example, these Ruminococci and Lachnospiraceae dominant clusters, they were then associated more with the responders. Also, Paramsopti and et al. They showed that there was a specific bacterial taxa that were associated with the clinical remission in inflammatory bowel disease. And for example, this Eubacterium halli, nowadays Anaerobutyrichum halli, and Roseburia inulinivorans were then increased in these patients that stayed in remission. And then something about the pouchitis. So all in all, these patients, if they have chronic pouchitis, they need to use a lot of antibiotics. They might use them recurrently or even continuously. So the microbiota is unstable anyway. And here, Selvik et al. Show that the response to the FMT is also very unstable, and the microbiota change is not that high in this patient group. They only found that one patient actually had this very nice change towards the donor microbiota. So there might be this individual response in this patient group, but also what is important is that the niche differences between the pouch and colon, because pouch and colon are very different environments. Pouch, for example, has more oxygen in it. So of course then different kind of bacteria likes to live there. So then something about the microbiome changes and the clinical effects. So majority of the studies that have been studying the microbiota changes in FMT has been concentrated on bacteria, of course, because that is the majority of the gut microbiota. However, we do know that, for example, bacteriophages, which are viruses of bacteria, and some eukaryotes, some, for example, fungi, they are also transferred in FMT, and they even might have association with the clinical outcomes. Then when we are transferring fecal material, we are also transferring other components than just the bacteria or other taxa. So we are also transferring the bacterial metabolites, host metabolites, antibodies, and other proteins. So that comes to the question that how the microbiota functions in the new environment, and should we also actually look into that. And we know that, for example, bile acid metabolism is disrupted in those patients that have recurrent C. difficile infection, and it is corrected then after FMT, and that has association with the clinical outcome, because that, for example, offers colonization resistance against the C. difficile. And there was a few years ago very nice papers from Smith et al. And Ioner et al. In Nature Medicine, where they actually looked into this strain level microbiome changes and could they be then associated with the clinical outcomes. And both of these papers concluded that the strain colonization is variable, and that, for example, the retreatments with antibiotics or the patient condition have effects on the strain colonization. And interestingly, this first study by Smith et al., they did not find an association with the strain engraftment and the clinical outcome, whereas this other Ioner et al., they actually found this connection between strain engraftment rate and clinical success. However, both of these studies included many indications in these analyses, and they also used partly the same data. So I think there is still questions to be asked whether the strain level engraftment has connection with the clinical outcomes. So as a conclusion, FMT is effective to modify the recipient's microbiota composition. Of course, the level, how much is engrafted, may vary between the different indications, and whether the colonization is necessary for a positive clinical outcome in indication beyond recurrent C. difficile infection is still under investigation. And as I mentioned, in addition to the taxonomical changes, also the functional changes matter, and they should be studied further, and also different components